Good morning.
Morning.
What did they say? Good morning, Hanover High. All right, so what do we got today, Mrs. Smith? Talking about DNA, right?
We are talking about DNA. It looks like we have Lauren in the house, Deanna, Libby, Alanis, Grace, Isaiah, that SOS Biology showing up there, Nolan, Keeney, Emma, Reef,
Miguel.
Is that it? Really?
I think you got everybody that said hi. So make sure you say hi in the chat. We'll give you a way to check in later for certain. But there's Caden.
There's some new people popping in there.
Ashlyn, Daniel,
Lena, Nicholas.
All right. So I think we get right into it because we want to kind of get through it. I'm going to give a little warning to everybody. We've been saying, like, done with DNA halfway through this week, start cell reproduction, because next Friday we will be going over cell reproduction. Mrs. Smith and I are talking about that a little bit. I don't want you to feel I'm giving you an extension here. That's not what we're doing. But we're trying to figure out maybe cell reproduction isn't the review to do next week. So we're going to be talking about that a little bit. So you might see some changes in that initial document. But because really, what do we got to go over before the test? We got to go over cell reproduction. We got to go over genetics. We got to go over ecology. And we got to go over evolution. We feel you have a strong evolution and ecology background, even if you don't know you do. But we feel that you do. But we'll talk more about that as we go. What I need from you right now in the chat, I need let's play versus. DNA versus RNA. I want you to give me one fact about DNA followed by one fact about RNA. Maybe a little versus in the middle. So this versus that kind of thing. Was that the form that you just sent me? Okay. All right. So in the chat, this versus that. We're not going to be here forever. Let's go. So deoxyribose, yes, Emma, but versus what? So put it together, maybe. Versus RNA ribose. So a ribose versus a deoxyribose, right? Or a deoxyribose versus a ribose. Deoxyribose sugar versus ribose sugar. Good. Double-stranded versus single-stranded. Good, Lauren. DNA first, Andrew. I know you know what you're talking about, but you put single versus double. Double versus single, right? If we're doing DNA first. DNA thymine, RNA uracil. Good, Miguel. Ashlyn, good. Double-stranded versus single-stranded.
Location.
Oh, location. Thymine versus uracil. Give another couple seconds. I'm waiting for the location one to pop up. Nobody's biting. All right, here's what I have. This is the actual graphic organizer we made last year or 2 years ago during reviews. Those videos are still around. We're going to share those with you, but I'm going to show you the top part of that right now. Give me 1 second. I'll get that top part up on the screen for you. All right. There's the top part. And I have DNA versus RNA at the top. So if you want to write this part of the graphic organizer down, you certainly can do that. You might just have to come back at the end, pause, and write it down. So I don't think it'll be up super long, but let's do it. DNA versus RNA. Double-stranded versus single-stranded. Deoxyribose versus ribose. Thymine, adenine, guanine, cytosine. Uracil, adenine, guanine, cytosine. A to T, C to G in DNA, A to U, C to G in RNA, right? This is all stuff you know. I thought this was an interesting one. Read 3 to 5. We read DNA 3 to 5, which means if we're writing RNA, we're writing it from 5 to 3 because it will run opposite. Stays in the nucleus. DNA stays in the nucleus. RNA leaves the nucleus. And I like leaves the nucleus better than found in the cytoplasm because it is made in the nucleus and it leaves. All right, and then there's multiple types of RNA, which we'll talk more about here in a minute. Is it worth mentioning the structure really quick? That it is a nucleotide, the monomer of DNA. We got to start using those words again for you, right? The monomer of DNA. The monomers that we put together to make the polymer DNA is a nucleotide, which is made of a sugar, an acid, and a base. Sugar, an acid, and a base. And that it runs anti-parallel to each other. All right. So one more thing while we got paper down here that I wanted to talk about before we jump into the questions. And I think the plan is to jump into the questions and then maybe play a blue kit. Is that right, Mrs. Smith? All right. So one more thing I want to talk about, and this will help you even on the DNA test you have coming up. I think it's really important that we mention or talk just a little bit. You can't see that. Can you see that? If I write right on? Yes, there we go. The central dogma. So this idea kind of confuses people, but it doesn't need to be confusing. It's just really looking at kind of this idea of how DNA works. It's a central dogma of DNA. A camera like goes out of focus and then it comes back in. We'll try and keep it focused for you as we go. All right. I want to start over here with a piece of DNA. So let's draw a piece of DNA. I'm going to try my best. Oh, not too bad. Done worse. These are our bases, right? So we could talk about replication, but we're not going to. We're talking about central dogma right now. But you do know that DNA replicates. You know that it's unzipped by helicase. So you know that new nucleotides are added and all that. Yeah, Nick, I'll work on the camera. So this is DNA. What's the first thing? So this is also in the nucleus. I'm going to make a little nucleus here. I'm going to make like a little nuclear membrane, right? So just so we remind ourselves that that's in the nucleus. What's the first process that when, let me think, how do I want to approach this with you? Let's do it like this. I'm going to make a single-stranded nucleic acid made out of nucleotides. What is this process called that's going to make a single-stranded nucleic acid? Anybody know? I'm sorry. You ask a question and then you got that 10 minutes of wait time. So let's. I'm waiting for that answer. Hey, ladies and gentlemen, if somebody wants to jump in the chat and try to disrupt it, that's cool. You just ignore them because we got things to do and we don't have time for that. Here we go. So what is that process? Yeah, well, replication, no. Replication, we're making a piece of DNA, so that would be a double-stranded nucleic acid. If I'm making a single-stranded nucleic acid, single-stranded nucleic acid, what am I making? What's the process? I'm making RNA then, right? So one of the things I always want to point out, if I write trans, I'm going to write it kind of weird, right? Transcription is what I was looking for. Thank you, Emma and Nicholas, to see it there. So transcription, we're going to make messenger RNA. So we're going to make, right now, messenger RNA. Why? To write a message. DNA cannot leave the nucleus. In order to get out of the nucleus, we got to send a message. When you can't leave a classroom and you want to send your friend a message, what do? You type a message and you send it. Bam. Right? Same thing. You can't leave the classroom. DNA can't leave the nucleus. It writes a message. Why did I highlight this? Why did I write script in big letters? Because if you think about when you're looking to change the text in a Google Doc, some of those fonts are called scripts. If you're thinking about a play, they hand you a script to learn. So it's a written thing, right? So I want you to keep that in mind. We're writing a message, messenger RNA, with a script. Okay, so that messenger RNA is going to leave the nucleus. It's going to go out into the cytoplasm. So I'm going to draw a little piece of RNA here, and I'm drawing it kind of weird, but just so we can get the idea. That piece of messenger RNA is going to link up with a ribosome. What's a ribosome made out of? I'm trying to hurry this along, so I'm not going to wait for the answer, but a ribosome is really, that's the pirate DNA. Or the pirate RNA, right? That's rRNA, ribosomal RNA. And I don't know that we've talked a whole lot about that, but a ribosome really is just RNA, right? So think about this. We make messenger RNA, leaves the nucleus, comes out to the cytoplasm, hooks up with ribosomal RNA or a ribosome. We're going to read that in groups of 3, and you should remember that as being a codon, right? And so we're going to match tRNA. So out here in the cytoplasm, we got these tRNAs floating around carrying amino acids, right? And I'm not going to get too specific here. We're just doing the general process, right? So they're floating around. They're going to come down. They're going to meet inside the ribosomal RNA and carry those amino acids. And as they do that, we're going to start to see a chain of amino acids form based on the order that we have those tRNAs going into the cell. You with me so far? So what is this process right here called? Where the mRNA is connected to the rRNA and the tRNAs. These are tRNAs, right? What is that process called?
Emma's got it. Andrew's got it. Translation.
Good. So we'll write that down. Am I still on screen? I'm off screen. Oh my gosh. This is getting, I'm running out of room to be, whatever here. All right, transcript translation. So just like when you send a message, right? Whoever receives that message has to read it and translate it into thought. If you send somebody a message in Spanish, they have to translate it in English maybe or whatever. All right, so what else did I need to do here? This forming right here, I think it's worth mentioning, is a poly, right? We're making little peptide bonds between all those amino acids. So it is a polypeptide. And that polypeptide will eventually fold and it will become a protein. So this idea of the central dogma is literally DNA to RNA to protein. That's it, right? Like that's how life works. DNA to RNA to protein. We have the code for life written in the DNA. We take that, we make it into a message, we send that out to the cell reads that, translates it, makes a protein.
And I had some students that were confusing transcription and translation because they're saying, well, it's going from DNA to RNA language. So we're like translating languages. Yes, but remember that both the DNA and RNA are both nucleotides and nucleic acids. So it's really the same language. It's just different letters. But then when we get to amino acids, we're changing to a completely different macromolecule and a completely different set of monomers. So if that helps you to remember and distinguish between transcription and translation too, that's a really good add-in.
Yeah, I like that because like right here, if you think about it right here, everything before that's nucleic acid. After that is protein. They're two different things. So how do we go from a nucleic acid to a protein? We need to translate it. That's good. Very nice. All right, so that's the central dogma. DNA to RNA, RNA to protein. And if you understand this, notice we left a lot of stuff out, right? We left matching the base pairs up and out and all of that. We left a whole bunch of stuff out. But if you understand this basic idea, DNA, transcribe it into RNA, translate it into protein, and where it happens, you're 90% of the way there. And 90% of the way there gets you there. You know what I mean? All right, so questions? Do you have that sheet up, Mrs. Smith? Can I pull it up?
I do have that sheet up.
All right, here we go. She's going through the questions.
Okay, so this first question in this section says, which statement describes a cell process that is common to both eukaryotic and prokaryotic cells? So all of a sudden you might go, wait a second, like we carried or talked about eukaryotic and prokaryotic cells the other day. Right. So this is the Keystone where you're going to be using information from everything. So as soon as I see that, I'm going to be pulling back out that graphic organizer I've already produced. That's a Venn diagram of prokaryotic and eukaryotic cells, and Mr. Suter's going to show you for a second. And so when I'm looking at that, I'm like, all right, so it has to be both. So it has to be something that talks about what is in both of those. So if I flip back to the question again, and, you know, kind of imagine, he can't show you both screens at the same time, unfortunately. But if we look at it, it says it's common to both. So question A says, or answer A says, both cell types carry out transcription in the nucleus. Well, prokaryotes don't have a nucleus, so it can't be A. So even if I get confused, I have this document right there that says, nope, can't be that. So A is out. Okay, if I flip to B, and B says both cells types use ribosomes to carry out translation. Okay, well, I know ribosomes is both, so that's a possibility. So I'm going to come back to that because I'm going to do the easy thing first and cross out the answers I know that it's not. I'm not worried about time because I can take as much time as I want on the Keystone. Question C says both cell types assemble amino acids to carry out transcription. Well, when I read that, well, amino acids don't carry out transcription. So like that one's out. And D says both cell types carry out translation in the endoplasmic reticulum. Well, translation occurs at the ribosomes in the endoplasmic reticulum. Well, even if I don't think about that, endoplasmic reticulum is not in prokaryotes. If it had said ribosomes, then I would have to deal with ribosomes. But endoplasmic reticulum is a membrane-bound organelle, and prokaryotes have no membrane-bound organelles. So the only possible answer on that one has to be B. Okay, both use ribosomes to carry out translation. Okay, so that is that first question there is answer B. All right, scooting on down to the next one. And this question's a little bit bigger, but I'm trying to show it all on the screen at the same time. Okay, the endoplasmic reticulum is a network of membranes within the cell and is often classified as rough or smooth, depending on whether there are ribosomes on its surface. Which statement best describes the role of rough endoplasmic reticulum in the cell? So it gives you a hint in there. It talks about ribosomes. So again, if you remember from the previous question that ribosomes are going to deal with translation, which means the production of proteins. So we're dealing with the production of proteins. So the first one says it stores proteins. Well, proteins aren't stored, so that one's out. It provides an attachment site for larger organelles. No. C, it aids in the production of membrane and secretory proteins. Well, potentially, you know, it's talking about proteins that are going to leave, and then it says it stores amino acids required for the production of all proteins. So this one, you're down to C and D, and I'm curious about this one, which one you think that it is, because when you're looking at this, you might be thinking it's either one. So in the chat real quick, C or D, does it aid in the production of membrane and secretory proteins? Or does it store amino acids required for the production of all proteins? I know I've talked about this to some students, but so this one you have to think about, where are the amino acids coming from? Where are the amino acids coming from? So far, perfect. Okay, so amino acids, remember, Mr. Suter talked about, are coming in from the cytoplasm on those transfer RNAs. They're not stored in the endoplasmic reticulum. So perfect. We're going to C. Great job. All right, moving down to question 32 on this one. It says a genetic mutation resulted in a change in the sequence of amino acids of a protein, but the function of the protein was not changed. As soon as I see there is a change, but the protein function was not changed, I have to think about, all right, which one doesn't change the function? And the easiest answer there for me is thinking, all right, silent mutation. So then I look at the bottom and I see silent mutation, nonsense, nonsense. Nonsense makes no sense. And I'm like laughing now because Nick sent this video to Mr. Suter and that's all I can think about at this particular second. But so nonsense is not going to form a protein that works. So I can quickly get it down to a 50-50 shot. So then I reread A and it says it was a silent mutation that caused a change in the DNA of the organism, or B, it was a silent mutation that caused a change in the phenotype of the organism. Now this one's tricky because we haven't talked about phenotypes yet unless you've hit genetics already, or you maybe remember from Mr. Beichler, or if you weren't in this school district, in a different one. So a silent mutation causes a change in the DNA, but not in the protein, which means it doesn't affect the phenotype. So it's going to be A. It is definitely A. All right, and then floating down to the last question here. This one talks about genetic engineering. So this is your biotech question. Genetic engineering has led to genetically modified plants that resist insect, pests, and bacteria and fungal infections. Which outcome would most likely be a reason why some scientists recommend caution in planting genetically modified plants? So unplanned ecosystem interactions, reduced pesticide and herbicide use, improved agricultural yield and profit, and increased genetic variation and diversity. Even if I wasn't sure about any of this, the first thing it says in the question is recommend caution. So if it's a benefit, it's not going to be something that they're going to caution about. So letter C says improved agricultural yield. That means more plants and more profit. C's out. D, increased genetic variation and diversity. Increased diversity is always better. More diversity means if there is a problem, that there's more variations and less likely going to result in extinction. So C and D are out. So now I'm down to unplanned ecosystem interactions or reduced pesticide and herbicide use. Well, I'm really not down to 2 because reduced pesticide, isn't that better? Doesn't that mean that I don't have to use as many pesticides that could affect the environment? So the best answer is A. Okay, so there you go with our questions. I'm pretty sure that's the last one on this one. Yep, for this one.
Awesome. Somebody asked earlier about what kind of score you needed to pass the Keystone, and the problem with that is nobody knows.
Gosh, I was trying to avoid that question, Mr. Suter.
Yeah, well, I mean, it's easy. It's just nobody knows. You know, once everybody takes the test, they figure out who passed.
Right. I mean, it's the score. The score is a 1,500. But
yeah, you just want to do good. You want to do your best. Bottom line, go into the test, do your best. That's all we ask you to do. If you go in and do your best based on doing your best all year, I'm not going to have any problem with whatever happens. Whatever happens. All right. So we playing Blue Kit. Do we want to do that for those that want to play? You have that?
Hi, I'm ready. All right, just got to share that screen again with you.
I'm up for a little game of Blue Kit on DNA. We'll see what happens.
Is this the right one?
That's like my jam too, right there. I tell you. Why people are signing into Blue Kit? I'm going to grab the link for that attendance form and post that too.
Yes. And yes, there are open-ended questions in the Keystone. There wasn't in this particular section that we went through today, but I will tell you, if I were thinking about what an open-ended question would be for DNA, remember this amino acid mRNA codon chart. I could see them asking you a question about using one of those and having to figure out, potential things as far as mutations. And remember, form is function. You change the form, you change the function.
The form is in the chat.
So we got 8 in. How long do we wait? Andrew said he needs to beat Ricardo, but I don't think Ricardo's here. So.
I just want to steal stuff from Grace. Hi, Grace.
No, you will never need to memorize the amino acids. Like they would always give you this chart. Even at the AP biology level, they would have to give a chart. I don't know them. Like I know a few off the top of my head, but no, absolutely not. Oh, we're up to 13. We ready then? And a 3, and a 2, and a 1.
Wait, we're
If you're
going?
pointing, the code's up at the top. You're playing, aren't you? Now I feel like I gotta play.
I was trying to, but it's not letting me sign in.
Really?
I'm at the enter nickname. Okay, there we go. Was waiting for a host. Oh, there we go. I don't know, but that music.
I can't get signed in either. I was gonna play, but I can't. And I like this blue kit because it it goes back to some of the enzyme names and stuff and like that we did not review. So
I don't know. Can you mute that? Mute that bloody
you don't like
music?
it? It's very relaxing. I don't know what you're talking about.
I can't even read a question with it blaring in my ear. It's genetic information. These questions are weird. I have to read them like 2 and 3 times.
Well, when you don't know the information, it can.
Oh, shit. I hate you.
Do you know it's gonna be May tomorrow?
Yeah.
And oh my gosh, I almost forgot. Is Keeney here? Keeney's here. I know what I gotta get ready. Somebody's birthday.
Oh, yes.
Oh, Ricardo is here. Only in your dream. I didn't even see that Ricardo was here. I can't get in like I've been trying. It doesn't like me today. Oh well. There's only 2 minutes and 21 seconds left. It's so quiet without the music.
Yeah. And I'm sitting here, I have to concentrate. These kids are killer. They steal all your money. And yeah, this one's pretty good. The numbers. What's this nothing? Now all of a sudden I'm not getting anything. It just says nothing for my gold.
What do you have, like 500 and they have like 0.2 million? 46,000.
Oh my gosh. Read it wrong.
It's not like, oh, there. I was like, why isn't it going over? Dun, 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 dun. Maya.
Are you kidding? I had that much money and I was still not in the top? All right.
Okay.
That's just the way it is, I guess.
Wow. I
Who
did.
won? Who won that one?
Maya. Maya won.
Maya? Maya who? Did it say? Was
No.
it Maya Brown or Maya Plank? Who was second? I'm sorry, I missed it.
Caden and Gray. All yours. I don't know.
Oh, wait. All three were my students? I don't feel so bad when I lose, but all three that won were my students.
Oh, no. It's going to be the rest of the week this way.
I just wanted to hear you say that.
Yeah, I'm sure you did.
All right. Well done, everybody. Maya Plank. Good job. Libby said she only missed one question. I only missed one too, Libby. I read it wrong. That was my excuse. All right. So do we need to do one more thing? Do I need to add it?
You need to add it.
3, 2, 1.
Happy birthday to you. Happy birthday to you. Happy birthday, happy birthday, happy birthday to you.
Whoa.
All right. Have a great day, everybody. Get caught up. Get through DNA. We'll see you.
I have Nick's video there if you want to.
Oh my gosh. Called a nonsense mutation. What is this even from? Right? Which kind of makes sense, right? Get it? Sense? Makes nonsense. It doesn't make any sense at all. Kind of makes sense. Does make sense.
There you go.
That was total nonsense.
Nick's little 30 seconds of fame.
Sentence form coming at you again. There it is. You better do it quick. We'll see you later. Have a great day. Love you.
So long.